Right, so today we're looking at viruses and bacteriophages. Let me put it on slideshow for you all. Right, so just some intro information about viruses. They are simple acellular entities. Acellular meaning that they are not composed of cells because they lack a similar structure to the way in which regular cells look. And they are not considered to be living things because they cannot survive or multiply on their own. They cannot reproduce on their own. They depend on a host organism, right? They consist of one or more molecules of either DNA or RNA that are enclosed in a coat of protein. So you can have RNA viruses or you can have DNA viruses. And they can be reproduced, as I mentioned, only within living cells. Therefore, they are termed obligate intracellular parasites, meaning they feed off a host cell in order for them to multiply, right? The nucleic acid strands that exist within these viruses can be either linear, closed cycle, or they can assume either of those shapes, linear or cyclic. Viruses are classified on the basis of their nucleic acid characteristic, their capsid symmetry, and the presence or absence of an envelope, their host, and other properties, right? Simply speaking, the structure of a virus is made up of a capsid and a core. A capsid is the outer coat of protein, while the core contains the genetic information that exists as either DNA or RNA. So quite simple, if you have to describe viral structure, you would say it's made up of a capsid and a core. The capsid is made up of proteins. It dictates the shape of the virus, and the core is either DNA or RNA. So viruses differ from living cells in at least 3 different ways. They have simple acellular organization, the absence of both DNA and RNA in the same virion, meaning you don't find both DNA and RNA in the same virus structure. You must have one or the other. And their inability to reproduce independently of cells and carry out cell division as you would have in regular cells like prokaryotes and eukaryotes. Viruses can exist in 2 different phases. They can either exist extracellularly or intracellularly. In the extracellular phase, they possess very few enzymes, and that is why they lack the ability to create copies of themselves on their own. Because of the fact that they lack those enzymes, they cannot make copies of their proteins as would be required in the process of protein synthesis. So they lack a lot of those synthesis enzymes that are needed for when we did transcription, translation, and so on, those enzymes that are required for the process of protein synthesis. So extracellularly, they lack those, sorry, I have someone. Okay. Yes, I see, no problem. Extracellularly, they lack those structures, and intracellularly, they exist as a replicating machine, basically, where they make copies of their nucleic acids because they use host structures and host enzymes and the host metabolism to synthesize virion components. And then they would take those components and build them together to create a complete virus particle. And then those viruses are released into the system. The particular host range of a virus is determined by the requirements of the virus. So if it needs specific enzymes that you would find in, let's say, plant species, it would be more than likely a plant virus because it would get those specific enzymes and requirements from that type of host. So that's why they are termed as host specific, right? They have specific attachment sites that are only present in particular types of host cells and the availability within the potential host of cellular factors that are required for viral multiplication. So depending on what they need to create new viral particles, that is how they would target their hosts. There are 3 main classes of viruses. We have animal viruses, bacterial viruses, which are viruses that would infect bacterial organisms, and those are called bacteriophages. And we have plant viruses. When we think about size, you can have various varying sizes of viruses. Although they are much smaller than bacteria, some of the larger viruses, for example, smallpox, can actually be about the same size as some very small bacteria like mycoplasmas and rickettsias and chlamydias, right? So very rarely, but the possibility does exist that viruses may have a similar kind of size to very small bacterial structures. Viruses generally range in size from 20 to 300 nanometers in diameter. We spoke about those different measurements when we talk about microorganisms. So you should have an idea, you'll understand what a nanometer means. So this diagram here would show us a comparative image of several different viruses and bacteria. So you're seeing there that the length of the screen averages at about 1 micrometer. So we're seeing there some of the extremely small viruses. You have your polymovirus, a bit larger is your influenza virus, your T. even coliphage. If you look at structure F, that is exactly what a bacteriophage looks like. Or those are the types of viruses that affect or infect the bacteria, I should say. And you have larger spherical structured viruses such as the herpes virus or the vaccinia virus. Those are larger in size compared to those such as influenza. So you see the wide range of structures as well as sizes that you can have of your viruses. Virus particles, also known as virions, may vary widely in size and shape. Viruses, as we know, are smaller than cells, ranging from 0.2 to 0.3 micrometers. The smallpox virus is one of the largest viruses that you would find. It is about 200 nanometers in diameter, whereas you have poliovirus, one of the smallest, is only 28 nanometers in diameter. So we have large variation in sizes. So we can have different types of genomes present in virions or viruses. You can have DNA viruses, and these types of viruses either would be single structured DNA, single stranded DNAs viruses, or double stranded DNA viruses. So you can have two types of DNA viruses. Similarly, you can have two types of RNA viruses, single stranded or double stranded RNA viruses. And in very rare cases, you will have RNA to DNA viruses, where they may convert, depending on what stages they are in, from an RNA virus to a DNA virus, meaning at one point in time they would possess RNA, and then at another stage in their cycle, whether it is when they go from an extracellular to an intracellular virus, they may be converted from an RNA virus to a DNA virus because their RNA is used to make DNA, right? The genomes of viruses can be composed of either RNA or DNA, and some use both as their genomic material at different stages in their life cycle. However, at any particular time, you are only going to find one of those genomic structures, DNA or RNA, located within the virus structure. This is an example of what a bacteriophage looks like. We'll talk about structure. We'll talk about how it infects a little later in the lecture. So a virus can either have DNA or RNA or never both. If you look at the structure here of this bacteriophage, you can see the tail fibers, the base plate, and the pin. So the pin is used, as you will see in the image to the left, the pin is used to inject itself into the cell membrane and cell wall of the bacteria. And then the head of the structure is where you would find your DNA or RNA. It would use the sheet, the tube and the sheet to inject that DNA or RNA into the host cell. Most viruses are too small to be seen under a light microscope. All viruses have DNA or RNA located in their core genome, and they are surrounded by a protein coat or a capsid. The combined viral genome and capsid is normally referred to as a nucleocapsid. So they're showing you here examples of a naked virus versus an enveloped virus. The naked virus, sometimes you may have viruses when they are now formed, you would usually call them a naked virus. When they bud off from a host cell, sometimes they take with them part of the cell membrane or the cell wall of the host cell, and they gain an envelope, right? So that capsid, that the viral genome, together with the capsid, when it gets a capsid envelope, sometimes it's referred to as a nucleocapsid. So depending on what the virus, what stage the virus is at, or what type of structure the virus may have, or if it has or has not infected a host cell before, it may or may not have an envelope present. Some viruses have complicated structures and they are referred to as complex viruses. So for example, pox viruses, where you cannot identify the capsid because they have several coats around the nucleic acid. And there are other bacteriophages that have capsids and additional structures within them. So there's still a lot of unknown viruses that have not been determined as yet, they have not determined the genome as yet. So for example, although we had coronavirus or COVID-19 around for a longer period of time, because of the fact that it may not have caused human infections, we were not aware of its exact structure until now. General morphology. Viruses may be classified into several morphological types on the basis of their capsid architecture as revealed by electron microscopy. As you know, we can only visualize viruses using an electron microscope, or you can use techniques called X-ray crystallography to determine what type of morphological features are present on a virus. There are some viruses, such as tobacco mosaic virus, may have helical symmetries with the capsid surrounding the RNA genome. Many viruses that infect bacteria or bacteriophages, they have a more complex structure. They are not helical or they are not cylindrical. They are not spherical, but they have a more complex structure where they have a head that contains the DNA and a complex capsid, and they also have tail fibers, as in the image we would have seen earlier. There are some animal viruses, such as the adenovirus. They have isometric symmetry and a DNA genome, and others, such as coronavirus, have complex capsids and an envelope with protruding proteins that surrounds an RNA genome. Historically speaking, some of the worst types of viruses that have ever been discovered to cause one of the most severe diseases in humans are RNA type viruses. So for example, coronavirus, they give the example of here. HIV/AIDS is also an RNA virus. So this is showing you some images of viral structure. So here we see a polyhedral structure versus a helical structure, right? Where the protein coat is made up of proteins. Slight mutations can actually create new strains of the virus. The capsid assists in protecting the genome that is present within. Capsomer proteins can also play important roles because they assist in allowing the virus to bind to specific sites on the host cell. So it allows for the virus to attach and infect host cells to allow multiplication to take place. We can have polyhedral viruses, right? So they give an example here. In this case, this polyhedral virus has about 20 triangular faces and 12 corners. The capsomers of each face form an equilateral triangle. An example of this type of structure you will find in adenoviruses or polioviruses. Examples of helical viruses. Again, you would have a helical structure that surrounds the inner core. You can find this type of structure in tobacco mosaic virus. Or you can have, generally speaking, although you have polyhedral and helical viruses that do exist, the most common types of structures that you will find in viruses known to man would be a bacteriophage structure or a spherical structure, as is seen in this image here. A is showing you a bacteriophage, which is considered an irregular structure for a virus. And then you have a spherical virus, which is your HIV, your human immunodeficiency virus, right? And the numbers are representing different structural components. Number 1 points to the capsids of the virus. 2 is your DNA or RNA. Well, 2 is DNA, sorry, in the case of the bacteriophage. 6 in your HIV virus is your RNA. 3 is showing you the envelope. Notice that the envelope is present. You would find enveloped type viruses usually only in spherical structured viruses, as well as envelope proteins. Those envelope proteins assist a lot in host infection because they allow for them to bind to, as I mentioned before, they allow them to bind to host binding sites more efficiently. That could speed up the rate of infection. With regards to bacteriophages, you don't find envelopes in them because they are so specific and they can very easily infect bacteriophages because of the way they are structured with those tail pins that allow them to inject, that increases the efficiency of their infection. So the capsid of viruses, when in spherical shape, you usually find that they are surrounded by envelopes, right? They may vary in shape. Sometimes you can have helical or polyhedral viruses enclosed by envelopes, and they are referred to as such, enveloped helical and enveloped polyhedral viruses. So we spoke about viral structure, we spoke about the capsid and core. All of these things can be used as important characteristics for viral classification. So one of the first ways they usually characterize viruses is based on what type of host they infect. So the nature of the host, animal, plant, bacterial, insect, or fungal infections. So that's why we would call different viruses like human viruses or plant viruses or specific types of animal viruses. Usually they are named according to their host, right? Nucleic acid characteristics is another way that you can characterize viruses, whether they have DNA or RNA, and if those exist as either single or double-stranded. They can also be named or characterized according to their shape or capsid symmetry, and they can be characterized based on if they have an envelope present. Other ways in which, so those four listed first are the four major ways that they are characterized. If you go into more detail on viral structure, you can also determine the diameter of the virion or nucleocapsid and characterize them in that way. You can count the number of capsomeres present on the envelope in icosahedral viruses. You can look at the immunologic properties, or you can look at the intracellular location of viral replication. So which types of cells are being targeted in the host to allow for viral replication? That's another characteristic you can look at when you are characterizing or trying to get information or study a virus in more detail. Now we're going to talk a bit about viral reproduction. In order, as I mentioned, in order for a virus to survive, it must first of all attach and be able to penetrate into an ideal host cell that possesses all of the different viral mechanisms as well as the important structures that are needed for viral replication. So they will target hosts that have all of the enzymes as well as all of the building components that they need to create new viral particles. So a virus must induce a living host cell to synthesize all of the components that are needed to make virus particles. These components must then be assembled into their proper structures to create new viruses or virions, and then the virus must be able to escape from the cell and infect other cells. So in some cases, you may have viruses infecting host cells, and they may lack the ability to lyse or to break free of a host cell so that they cannot spread. So sometimes organisms can fight off viruses in that way because they do not allow for the new viral particles to escape and cause new infections, right? The phases of the replication process can be characterized in 7 steps. In this diagram here, it's showing you a summary of the 7 steps of viral infection. We have attachment, penetration, early replication. Replication is completed, synthesis of proteins, assembly, and then release. We'll go through this process in a little more detail as we go up. Right. So this image gives you a better kind of idea as to what's going on with bacteriophage multiplication, and it shows you the step-by-step procedure that's taking place, right? So in the first step there, this is showing you a bacteriophage that is infecting a bacterial cell. So in the first step there, you have attachment of the phage. It's infecting E. coli in this case, and it injects the phage chromosome, whether it's DNA or RNA, it injects it into the host cell here, which is E. coli. Then in the second stage, there's breakdown of bacterial chromosomes by phage-specific enzymes. So the chromosomes are broken down to allow the process of protein synthesis to start off. So after the bacterial chromosome has been broken down, it allows replication of the phage chromosomes using the bacterial materials and phage enzymes. So notice here, the phage or the virus first of all destroyed the bacterial chromosome so that after that point, when replication of its DNA and RNA is being done, none of it is going to be wasted on the bacteria itself because it has already killed off the bacterial genome, right? So now it basically has taken over the cell, and any kind of type of replication and synthesis from this point on is going to be for the purpose of viral replication. So in the third step, you have replication of the phage chromosome using the bacterial materials and phage enzymes. In the fourth stage, you start having expression of phage genes that is going to produce the structural components of the phage or the virus, and they start assembling in the fifth stage to form new viral particles. And in the last stage, there's release of progeny of those new phages by lysis of the bacterial cell wall, and those new viral particles are released into circulation or released to go and now infect more E. coli bacteria and create more viral particles. Any RNA in the biosynthesis of viral components, any RNA that is transcribed in the cell is the mRNA that is transcribed from phage for the DNA by biosynthesis of phage enzymes and capsid protein, so that basically all of the host cell structures and host cell mechanisms are no longer being used for the cell itself, but it is used for the biosynthesis of viral components only. As I mentioned, it takes over that cell, right? During the maturation and release phase, the phage heads and tails are separately assembled from protein subunits, and then it begins to package. The head is packaged with phage DNA, and then the tail is attached to create that structure, right? So in this diagram here, they're showing you how the assembly takes place. They start off as individual proteins. Then you have the tail and the capsid, and they attach to each other. The tail fibers attach to form a final bacteriophage particle in its mature state, which is then released. Lysozyme, which is the enzyme that is used, provided by the phage gene, is synthesized as well. And this enzyme causes the breakdown of the bacterial cell wall, and the newly produced bacteriophages are then released from the host cell. The number of newly synthesized phage particles can range from about 50 to 200 structures. So when you have viral replication taking place, there are two different types of cycles that can be followed. There's lytic cycles and lysogenic cycles of viral replication. So virulent phages typically lead to the death of a cell through cell lysis. There are other types of phages, temperate phages on the other hand. They can end up being part of a host chromosome and are replicated with the cell genome until such a time that they are induced to make newly assembled viruses or progeny viruses. So you have two different types. The first one there refers to the lytic type of cycle, and the second description there refers to a lysogenic cycle, which I'll describe a little more, and there's a little video to show you at the end of the descriptive process, right? So what happens in lytic cycles? That is what we basically just went through, those stages that we talked about in the bacteriophage, where during the lytic cycle, you have attachment, penetration, it takes over the cell, allows for biosynthesis of new phage particles. They would structurally come together and form mature bacteriophages, and then they lyse or they break through the cell walls and they release new phages. That's what happens in a lytic cycle. In a lysogenic cycle, it's a little different, where the phage genome also enters the cell through attachment and penetration. Example, a Lambda phage cycle, and instead of killing the host cell, the phage genome integrates into the bacterial chromosome and it becomes part of the host cell, and it's now referred to as a prophage. A bacterial host with a prophage is called a lysogen, and this process is known as the lysogenic cycle. This usually happens in temperate phages and allows a virus to be latent or undetected, right? Whenever the virus is ready now to start to continue its replication and assembly and so on, it would then allow for the DNA of the virus to be created, and it allows assembly, and the lysogenic cycle may allow for the progression into a lytic cycle where it then continues to multiply and it would break free of the cell, right? The presence of the phage may alter the phenotype of the bacterium since it brings in extra genes, so it's integrating its RNA or DNA into the DNA or RNA of the bacteria itself. And then it changes the phenotype, the genotype of the host, and therefore also changes the phenotype. You'll remember, if you remember from genetics, the difference between genotype and phenotype. Genotype is a difference in the genetic composition, and phenotype is how it is expressed. So this bacteria will actually form a new type of bacteria, which may be more virulent. So Viruses can even assist bacteria in becoming more infectious or becoming more virulent based on a lysogenic cycle type of structure, right? So this change in this bacteria or host is called a lysogenic conversion or a phage conversion, right? This cycle here, the diagram here is showing us what's taking place. Infection, the phage DNA becomes incorporated. It does not break down the host cell chromosome. It integrates its chromosomes, its phage chromosomes, into the chromosomes of the bacterial cell itself. The cell then divides, causing this DNA or this prophage structure, prophage DNA, to be passed on to all of the daughter cells. Under stressful conditions, the prophage DNA may be excised, and then the bacteria will now enter into a lytic cycle where it starts to multiply to create. So before, at the top 4 stages, you can have a virus present in our bacteria in a latent state. And at any point in time where stressful conditions are endured and the bacteria is now going to get rid of that phage chromosome, release it from its structure, and then the lytic cycle will be entered where they will break down the chromosome of the bacteria and they will only start multiplying and creating viral chromosome structures and viral proteins and capsid and cores to create new phage particles. The phage particles will become assembled. The cell will then lyse and release newly made phages. So let me play this video for you, which should hopefully clear up any type of confusion between those two types of cycles.
Hey guys, how you doing? This is a video to show you the difference between the lytic and lysogenic cycles of how bacteriophages can reproduce. Just remember that this process, or both these processes, have only to do with bacteriophages, which is viruses that attack bacteria. It doesn't happen in animal viruses like the flu and other things. So basically, to kind of give you a sense of what's going on here, we see that here we have a bacteria. So this is the bacteria right here, and this is the chromosome of the bacteria, and this would be a bacteriophage. This is the virus that attacks the bacteria. So if we kind of see what's going to go on here, this particular virus is going to, as you can see, inject its DNA into the actual bacteria. And so now this bacteria has its own DNA, and it has the DNA from the actual bacteria. And so if we see what happens from this point on, we'll now see that the host DNA has been destroyed. The base of the phage DNA, the genes in the phage DNA, directed the host DNA to fall apart. And so now the only DNA that's left in the actual bacteria is the DNA from the virus, and I'm sure you can figure out what's going to happen from this point on. Now, of course, the ribosomes of the bacteria are going to be producing proteins. These proteins are viral proteins, and as you can tell here, the phage DNA is going to now code for the production of more viruses, and of course now these viruses are going to at some point explode from the cell, and that is basically the lytic cycle. And of course lytic means to lyse, or it means to cut or break open. So this results in the destruction of the bacteria, and that is basically the lytic cycle. So now we're going to go through the lysogenic cycle. So now this is the lysogenic cycle, and this differs in a couple ways, and I'll talk about them after. So the first part of this is the same, that basically a virus is going to attack the bacteria, and so what they didn't show here is that the virus would have landed here, it would have incorporated its DNA exactly the same as it did in the lytic cycle, but here's now the difference. In the lysogenic cycle, we see that instead of the DNA from the virus destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now the difference is that, there is no destruction yet. So the beautiful thing for the virus is that, well, basically, it's not going to be making a lot of its babies, for lack of a better word, or offspring immediately, but this cell is going to start to divide, and it's going to divide zillions of times, and every time it divides, you can see that little teeny piece of DNA from the phage is going to, it's basically referred to as prophage DNA, is going to be carried from generation to generation. So now you have potentially millions, if not hundreds of millions of bacteria that all have this little piece of viral DNA. So at some point, when this host cell becomes either stressed or exposed to ultraviolet light or some kind of chemicals, a system called phage induction happens. And basically what happens, you'll see here that that little piece of viral DNA pops out, and now basically the cell goes through the lytic cycle. So the second half of the lysogenic cycle is basically the lytic cycle. So what's going to happen? It's going to destroy the host DNA, it's going to start making the proteins, and there we go. So the difference between these systems is that in the lysogenic cycle, the chromosome or the nuclear DNA material from the virus incorporates into the actual phage DNA and is carried generation to generation. So the bacteria doesn't make it, or the virus does not make as many offspring as quickly, but it is incorporated its DNA into many, many more cells, and thus giving the ability to potentially make much more offspring over a longer period of time. And so that is basically the difference between the lytic and lysogenic, and I hope you enjoyed it.
Right. So we continue on. I'll take any questions after when we finished up with the lecture. Right, so that leads us now, after we understood how the cell cycle, I shouldn't say cell cycle, how the viral cycle takes place either in a lytic or a lysogenic cycle. Now we're talking about viral growth curves, right? So viral growth curves differ a bit from bacterial curves, which we looked at early on in the semester. Unlike the growth curve for a bacterial population, the one for a virus population over its life cycle does not follow a sigmoidal curve. So it has a different type of shape, right? Shape or structure. During the initial stages, an inoculum of a virus causes infection. Then it enters an eclipse phase where viruses bind and penetrate the cells, and no variants are detected in the medium. So you don't have any increases in numbers during that phase. The chief difference is in the next stage, the viral growth curve, you have release of virions occurring, and it lyses the host cell at the same time. And during this process, you have, sorry, some of the lectures cut off there. Let me just fix that. Right. That's right. So when there's release of or lysis of the host cell and release of the viral particles, there's a burst. So the numbers increase significantly in that period of time, and it's called a burst size. In this type of curve, you would have your host cell, when it becomes lysed, it releases as many viral particles into the medium as present, and it causes a steep rise in the number of viral particles. So you're going to see basically the number of viral particles going from 0 to 100 during that process, right? If no viable host cells remain, the viral particles will begin to degrade during the decline of the culture. So this is what the growth curve of a virus looks like compared to that of a bacteria. So you have 4 different stages. The inoculation stage, where the virus binds to the cell. Then you have an eclipse stage. That is the point by which you have all of the viral particles are present inside of the host cell. They are going through the process by which they are multiplying using the host mechanisms and the host structures to allow for the creation of new viral particles and assembly of new viral particles. When it lyses the bacterial cell or the host cell, it releases all of those viral particles. So you see the numbers skyrocket. And then it reaches a maximum or a burst size where the number of virions released per bacterium is recorded. And unless it's going to start over again to enter into new host cells and replicate more, it will then decline based on the type of infection that is taking place, right? Okay, no problem, Sydney. Right. Most bacterial viruses that have been studied in detail, that have been studied in detail, that should be, they infect bacteria of the enteric group, such as E. coli and Salmonella and so on. These are ones that have you have most types of information on and have been studied in a lot of detail. Viruses are, however, known to infect a variety of prokaryotes, both archaea and bacteria. A few viruses have lipid envelopes, most do not. In many bacterial viruses, you will also find general structures of the complex head and tail structure that we looked at, which is the general way in which a bacteriophage looks. Right? Head and tail structure. So generally speaking, when we talk about bacteriophages, they all have this common type of structure. They very rarely vary from what this looks like, right? The tail components assist in attachment of the virion to the host cell. Nucleic head is measured at about 85 nanometers in diameter, generally speaking, right? So that kind of leads us to tallying up talking about all of the different characteristics and understanding the information of how, with regards to how viruses grow and multiply, and understanding the viral growth curve, right? And the last part of the lecture, we're going to talk about viruses and the different types of infections that they cause. So viruses and cancer. A number of animal viruses can participate in the change of a cell from a normal one to a cancer or tumor cell. It's where you have uncontrolled growth taking place. Most cells in mature animals, although alive, do not divide extensively. Because these cancerous cells have fewer growth requirements, they grow profusely, leading to the formation of very large masses of cells called tumors. So because of the amount of structures needed being less than that of a general viral particle, the multiplication of these types of cancer cells actually happen much faster, so tumor growth occurs very quickly. Generally speaking, normal cells, the development of cancer is a multi-step process. It occurs usually inclusive of mutations arising either spontaneously or as a result of exposure to different chemicals like carcinogenic material, or by physical stimuli, exposure to things such as UV radiation or X-rays. Certain viruses can also bring about a genetic change that can result in the initiation of tumor formation. So when we talk about cancer cells, we generally think of it usually being formed by exposure to different types of carcinogenic materials. However, because viruses can cause mutations and genetic changes that dictate or replicate some of these types of exposures, they can also initiate tumor formation. These are some examples of some human cancers where you have viruses playing a big role in them. Example, T-cell leukemia. This is caused by a human T-cell leukemia virus. Burkitt's lymphoma caused by the Epstein-Barr virus. You also have nasopharyngeal carcinomas caused by that type of virus. The hepatitis B virus can cause hepatocellular carcinomas or liver cancer. And the papilloma virus can cause skin and cervical cancers. Then you can have pox viruses, which are the most complex and largest type of animal viruses. They are generally not able to metabolize and they depend on host machinery for protein synthesis. And they are unique in that they are DNA viruses that replicate in the cytoplasm of these cells. Smallpox was one of the first type of pox virus to be studied in any detail. And it was the first virus for which a vaccine was developed. So study of pox viruses and the history of diseases that they have caused allowed scientists to understand a lot about viruses and their replication in order for them to create vaccines, especially because the pox viruses were so large, it was easier for them to be studied. They can actually be seen under really good light microscopes. For senior virions, these types of virions are taken up via phagocytic processes that allows the cores to be liberated into the cytoplasm for them to be multiplied. You have another group of viruses, adenoviruses. These consist of linear double-stranded DNA of about 36 kilobase pairs. They must create protein codes that are essential for the infective properties of their DNA. The DNA is generally about 5 to 6 to 70 times of the size of other DNA papoviruses. This gives a bit more information about how the replication process of these types of viruses takes place. We're not going to go into any type of too much detail. You don't need to know about extreme detail of those, right? We're just talking about different categories of viruses. We have retroviruses. They are RNA viruses, but they immediately use their RNA to create DNA structures as an intermediate using the enzyme reverse transcriptase. They were the first types of viruses shown to cause cancer and have been studied in most extensively because of their carcinogenic characteristics, right? Example, AIDS, the one that causes the acquired immunodeficiency syndrome. They were the first type of viruses, as mentioned, to cause cancer, and therefore most of the studies have been done on these types of viruses. And the enzyme used here, reverse transcriptase, has become a major tool in genetic engineering because they have tried to manipulate these types of viruses to prevent the conversion of the RNA to DNA in order to control the spread and to stop their infective properties, right? Viroids and prions are structures, well, viroids, as we know, are small circular. This is another term that is used for viruses, right? Sorry, virions are another term used for description of viruses. Virioids are similar to the structure of viruses. They are small circular single-stranded RNA molecules, and they can act as pathogens. They have an extracellular form where they lack a capsid, meaning they just exist as a naked strand of RNA, right? Which is extremely interesting because you have no protein code encoding genes. They just copy the RNA to create new virioid particles. Prions, on the other hand, they are the other extreme of virioids, meaning they don't have RNA or DNA. They are just entirely a protein structure. And they have been found to also create diseases in living things, right? There is, so the prion protein particle is infectious, and there are various prions known to cause a variety of diseases in animals. And then you have plant viruses, example, tobacco mosaic virus. You can read up on that, right? What I want to show you all, so as we're coming to the end of the lecture, what I want to show you all is on eClassroom. I have up some activities for you all to do. So if you all can go and do that now, that'll be ideal, right? So you could spend a little time finishing that unless you want to do it later on in the week. So if you see my screen here, you have the lecture up. I'm going to put up the recorded lecture that we did as well onto that.